In this video, I'll explain you the process of DNA fingerprinting. First of all, let's understand history and the basic of DNA fingerprinting. This DNA fingerprinting was first of all introduced by Sir Alec Jeffreys in the year 1984. He is a British geneticist who introduced DNA fingerprinting. He developed this technique. Later, Dr. Lalji Singh had introduced it in India in 1988. So this was introduced in India in this year by Dr. Lalji Singh. So if it is asked who is the father of DNA fingerprinting, Sir Alec Jeffreys, and who is the father of DNA fingerprinting in India, Dr. Lalji Singh. Now, what was the need of developing this technique? The first reason was for criminal detection. Suppose a person is murdered and there are two suspects A and B who might have murdered the victim. Then DNA from the victim is matched with A and B, and whoever person's DNA matches, that is the murderer. So one was to find out the criminal. That was the reason that DNA fingerprinting was developed. Second was paternity disputes. Sometimes a couple has dispute that the child belongs to some other person and not the father. Then if such a dispute is there, then the DNA is collected from the child and it is matched with the father and the person that the father claims to be the father. Then whoever person's DNA matches, that is the father of the child. So that was one reason that DNA fingerprinting was developed. And the third was genetic diversity. To find out the immigration dispute. Sometimes like the genetic makeup of your person from Europe or from Asia or from US, everyone has different. So when we study DNA fingerprinting, when we study the DNA of those people, we can understand from where that person must have migrated. So migration disputes are solved by DNA fingerprinting. Now let's understand what is the definition of DNA fingerprinting. It is a technique to identify a person on the basis of his or her DNA specificity. Every individual has a specific DNA, whether they are siblings, then also they have specific DNA, and that is why this technique was developed to explain the identity of a person. So it is actually a technique, it is actually a laboratory test or a chemical test which is used to identify a person on the basis of his DNA. Now let's understand what is the basic principle that is used in DNA fingerprinting. It involves identifying the differences in some specific regions in DNA sequences, and these are repetitive DNA sequences. So what is the difference in the repetitive DNA segment that is identified by DNA fingerprinting? Let's understand it more. Suppose this is a DNA molecule. We know that 99.9% of DNA is same in every individual. There is no difference at all. But there is only 0.1 difference, 0.1%, where the DNA is different. Here DNA is different. This 0.1 difference, 0.1% difference in this particular segment or in this area, DNA has repetitive sequences. That is, suppose we consider this area, it has A, G, A, C, A, G, A, C repeated many times. This particular area, because it has repetitive DNA sequences, it is called repetitive DNA. So wherever, if we are comparing two individuals, we consider the differences, we find out the differences in these repetitive DNA, what is the variation, on that basis the identity is detected. This area where these repetitive DNA is present, this is known as satellite DNA. So satellite DNA is this 0.1% of DNA which is different in every individual. Now there are two types of satellite DNA, microsatellite and mini satellite. Microsatellite DNA are AT rich, that is they have more adenine and thymine, and they have base pairs which varies from 2 to 6 base pairs in one repeat. So, if we consider that it has A, T, C, again A, T, C. So, suppose I am saying that there are 3 base pairs which are constantly repeated, then this is a microsatellite which has from 2 to 6 base pairs and which are rich in adenine and thymine. Now, these microsatellites, that is these repeats which are from 2 to 6 base pairs, they can be from 10,000 to 100,000 times. So, this can be how many number of repetitive units are present if microsatellites are present. If we consider mini satellites are those which are GC rich, means they have more of guanine and cytosine, and they have 10 to 100 base pairs per repeat. So, if we suppose consider that there is GGG, CAT, GG, CCA. It has more of G and C, and we count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. These are 11 base pairs. If they are again repeated, means again we go to G, G and so on, then these are mini satellites. These mini satellites range from 500 to 30,000 times. So, again satellite DNA is divided into microsatellite or mini satellite based on these 3 points. Base composition, if they are more rich in adenine and thymine, then they are microsatellites. If they are rich in guanine cytosines, then they are mini satellites. On the base of number of repetitive units, how many times the units are repeated, 10,000 to 100,000 times, then it is microsatellite, and 500 to 30,000 times, then it is mini satellite. And length of the segment, if it is just 2 to 6 base pairs, then it is microsatellite, and if it is 10 to 100 base pairs, then it is mini satellite. In DNA fingerprinting, we consider mini satellites. I already mentioned that when we do DNA fingerprinting, then mini satellites are considered. Mini satellites are those smaller portion of DNA where repetitive sequences are present. It is also called VNTR, or we can say variable number of tandem repeats. This is the full form of VNTR. It means the repetitive units are present multiple times. Now, if we consider this entire DNA molecule, which I have already discussed earlier, but I'll repeat, then the major portion of DNA, which is called bulk DNA, which is 99% of DNA, is same in all the individuals. There is no difference in this bulk DNA, whereas a small portion of DNA is called satellite DNA, which is only 0.1%, where the main difference is present, where we can identify different individuals based on the differences present in this DNA, and this is known as satellite DNA. So, we are considering mini satellite which is present in this area where the repetitive sequences are present, and here is the difference, that is there are variable number of these repeats which are present in this area. Now, if we try to understand the main process, that how DNA fingerprinting is done, then there are 6 steps. The first one is isolation of DNA. So, what happens is, if suppose there is a person who is murdered and there are 2 suspects, person A and B. So, we do not know who has murdered the victim, but there are 2 suspects. So, first of all, from the crime scene, the sample is collected. Now, the sample can be blood, it can be saliva, hair follicle, skin, any part of the body, wherever the cells are present, they can be extracted. So, from the crime scene, we take the samples and DNA is isolated. So, here from the cell, DNA is isolated. After that, it is digested by endonuclease enzymes. There are certain kind of enzymes which are called restriction endonuclease enzymes, and if we treat this DNA with this particular enzyme, restriction endonuclease enzyme, which are of various kinds, Then they cut this DNA at specific points and we get small fragments of DNA. Next step is separation of DNA fragments by electrophoresis. So before we understand that, what is done is from the crime scene, the sample is collected and the DNA is treated like that. First of all, it is extracted from the cell from wherever sample we have taken, then DNA is extracted and then it is treated with restriction endonuclease enzymes by which these are broken into fragments. Same is done with the sample of person A and person B, because we want to match the sample from the crime scene with person A or B. It means who is actual murderer of that person. Now we move to the third step, that how this electrophoresis is done. So, the electrophoresis is done by a method which is called gel electrophoresis. It is called gel electrophoresis because we use agarose gel in it. Electrophoresis is done in a chamber which is an electrophoretic chamber or electrophoretic capillary, and it has various wells. So, these are the wells. So, I have just given an example of like 3 wells. These wells are the places where the sample is put. So, if suppose we have collected the blood sample from the crime scene and that sample is C, we will place it here in one of the wells, and there are 2 suspects who can be the murderers. Their sample is also collected and it is filled in these 2 wells. After they are filled in these wells, they are treated with alkaline solution. The reason is that DNA is double-stranded molecule. When we place them in alkaline solution, it denatures. Denatures means the hydrogen bond breaks and these DNA molecules then they become single-stranded. We already know that they are broken into fragments by restriction endonuclease enzymes and now they have become single-stranded. Because of restriction endonuclease enzyme, these fragments will be broken at different lengths depending where they are breaking on the property of restriction endonuclease enzyme, because there are various kinds of restriction endonuclease enzymes and they cut at specific points. After that, we will start the electric current. DNA is negatively charged and here these wells where it is placed, this is cathode. So, because DNA is negatively charged, when electric current flows, then it will move towards anode which is positively charged. That DNA fragment which has less number of repetitive DNA sequences will move farther because it is smaller. This gel, agarose gel, will act as molecular sieve and it will allow the smaller fragment to pass more quickly as compared to the larger fragment, which has more number of tandem repeats or more number of repetitive DNA sequences. So, if we suppose that this fragment comes and stops here and a larger fragment stops here for crime scene sample, and then same for B also, and for A it stops at somewhere here and somewhere here. At this moment on the agarose gel, wherever these DNA strands are stopping, the scientists cannot see this because they are transparent, they cannot be seen. So, this is just to show you that we are assuming that they stopped at this point. This is gel electrophoresis, that is, we are running the current. These single stranded DNA strands, they will start moving towards the anode, and when the electric current flows, the smaller fragment with less number of repetitive DNA sequences will first reach towards anode. So, it will be farther, and whereas the larger pieces will slowly move towards anode. So, they will be at the lower end. After this is done, then the fourth step arises, that is, this is the third step that we have finished, that is separation of DNA fragments by electrophoresis. The fourth step is southern blotting, which is done by nitrocellulose or nylon membrane. So, what is done is that after this gel electrophoresis occurs, then a nitrocellulose membrane or a nylon membrane is placed on this electrophoretic gel. Why is it placed? So that these DNA strands which had stopped at a particular point on the agarose gel, they will be taken up by the nylon sheet. So this transferring of the DNA strands at the fixed point, the same point, from agarose gel to the nylon sheet or the nitrocellulose sheet is known as southern blotting. So southern blotting is just transferring of those DNA strands at the exact point from agarose gel to the nylon sheet. So after this southern blotting is done, then goes the next step, which is hybridization using labeled VNTR probe. Labeled means these are radio labeled, they are radioactive, and these are VNTR probes. Now what is a probe? Probe is a small segment of single stranded DNA artificially prepared which has VNTR, that is variable number of tandem repeats. So they have repetitive DNA sequences, they are small single stranded. For example, now if we consider that suppose there is the sample has CG repeats. So this is the tandem repeat, okay, CG. What is the complementary strand? GC. So these VNTR probes may contain, we can use this kind of probe which has GC repeats. These VNTRs are single stranded. Now what happens is after southern blotting is done, after those DNA strands are already attached on that particular exact point from agarose gel to the nylon sheet, then these probes are added to the nylon sheet. These probes have fixed number of tandem repeats. What will they do? Wherever they will find the complementary strand, they will form hydrogen bond and they will hybridize. Hybridization means they are forming hydrogen bond with their complementary DNA strands. So they will find their particular location wherever these complementary bases are present, and they will form hydrogen bonds and hybridization will occur. Hybridization means joining of these VNTR probes to that complementary DNA strand sequences. Last is detection of hybridized DNA fragments by autoradiography. So X-rays are passed through this nylon sheet and then wherever the DNA fragments, wherever those probes had hybridized with the DNA fragments, they will be detected. So now after that, the scientists can see those positions where the DNA strand had stopped, like for crime scene and individual B, and here the crime scene, this part and this DNA strand, wherever they stopped was the same. So it can be then concluded that individual B is actually the murderer. So this is the entire process of DNA fingerprinting, where these 6 steps are done, that is isolation of DNA. DNA is first of all extracted from the cell, from the crime scene or from the individual, wherever we are collecting the DNA sample. Then they are digested by using restriction endonuclease enzyme, where they are broken into fragments at specific point. Then separation of these DNA fragments is done by electrophoresis, and then these separated DNA fragments, then they are transferred onto a nitrocellulose or nylon membrane using the method of southern blotting. After that, the VNTR probes are added so that hybridization occurs, and then these hybridized DNA fragments are detected using X-rays by autoradiography. So this is the entire process of DNA fingerprinting. I hope you have understood it. If you like my videos, then do subscribe to my channel, save and follow for more. Thanks for watching.